Hi guys, Dr. Zveo. So what we're going to do in this quick little lesson is to better visualize what nature looks like at the molecular level. And the reason why I'm going back to this today is because we've been talking a lot about how we can inherit the necessary hereditary features that allow us to be who we are, to look the way that we look. And we're trying to better understand that process, to understand what makes us different from our siblings, and ultimately to explain the question as to how these two individuals can be twins yet look very, very different, right? And in an entry card last week, I noticed that still people are getting confused by oxidation, reduction, and electronegativity. And so I thought of a way to try to build a better model in your mind about this particulate nature of matter, because without that appreciation, it becomes very difficult to explain how genes are in fact matter in the sense that they are DNA and that they have specific chemical instructions. Really understanding what happens when those mutate to make changes in an individual and ultimately possibly in the species itself lies in its particulate nature, how those genes are actually just really a collection of atoms. And so what I want to answer this question is, how does the particulate model of matter or of nature help us to visualize emergent properties? And so when I close my eyes and I think about what an atom looks like, what a molecule looks like, all I see billiard balls. And in fact, I want you to close your eyes and I want you to imagine billiard balls. I want you to imagine those balls collide with each other. And really, it's the collision of these particles, and we call them atoms, of course, that lead to new combinations of those atoms, right? And when we visualize those, sometimes we approximate what that new physical thing looks like. We'll make that circle big and it'll call C. So A, due to its chemical properties, A and B will combine in very specific ways. Chemical properties will determine the reactivity. Will A react with B? How will it react with B? And there are, in fact, lots of patterns that you will delve into much more intensely next year in chemistry. But it suffices to say that they do combine. They form this new macro particle. They form A to B. They're now stuck together somehow. We'll talk about that in a second. But A and B have physical properties. And these physical properties, in fact, contribute to the new chemical and physical properties of this new macro particle C. Can we predict, can we infer that C exists by studying C? We can. So we study how C is, what this new macroparticle is, by really just studying a huge collection of them. So for instance, if this is sugar and we put it into water, we can study the physical and chemical properties of that new solution of sugar water, right? And we've done that so far in the lab on tonicity when we're talking about hypotonic and hypertonic solutions. But what influences these properties? Really, it's the arrangements of protons, neutrons, and electrons. Of course, more protons and neutrons will increase the mass of C. So that will also increase the density of C. So the amount of mass in a particular volume will increase as we increase the protons and the neutrons. And then also, if we have an unequal number of protons and electrons, this will also change the physical property of C. It can in fact change its charge. Okay, the more electrons there are, the more negatively charged C will be. The fewer the electrons, the more positive charge this will be. And the positive and negative charges, of course, if you have different particles, some are positive, some are negative, they will attract to each other or they will repel each other. So really, the fundamental nature of molecules, the density, the mass, and the charge can be affected by even smaller particles, those protons and those electrons that make it up. And it continues going outward and onward, right? New combinations of C with D and with E and with all these different atoms and molecules. Really, the combination of all of these physical properties can affect it. And we know that to be true because as I bang my table, I can sense that the particle is going to be different than if I sit on my comfortable reclining chair, all due to the combination of protons, neutrons, and electrons. Okay, so what then influences its chemical properties? Remember that valence electrons, the electrons in the outer shell surrounding the nucleus, will determine chemical properties. And the closer an atom gets to 8 electrons, that's supposed to be an 8 there, the more reactive it is. So how are they held together? Well, let's look at that. Particles are held together in different ways, and for each way, the new macroparticle will have a distinct set of properties. So recall, our particle C is really just a combination of A and B. But we have to ask ourselves, how are they held together? We know that there are ionic bonds, and we also know that there are covalent bonds. We draw them differently. We draw them differently because they are held together differently. If they're ionic, you have particle A being positively charged and particle B being negative. They're held together very tightly by the electrostatic attraction of these opposite charges. This sort of substance will have much different set of physical properties than if A and B were held together by a covalent bond or the sharing of electrons. So even though A and B can be held together, if they're held differently, their physical properties and, of course, their chemical properties will be very difficult. They're not going to be equal. Okay, so let's look at covalent. If A is more electronegative than B, and electronegative, of course, is the property of an atom that makes it hold onto those electrons more tightly when bonded to another atom. Electronegativity is a property. Okay, and the more electronegative you are, the more you're going to want to hold on to an electron in a covalent bond. And if you recall, we drew that as an electron going around A. In fact, that's what it does. The electron is shared, so it has to be moving between them. Okay, and so the more it's around A, the more negative it is, the more partial negative it is. And the more it's around B, the more because B has less negative charge around it, because the electrons spend more time here, it's going to be slightly positive relative to A. Okay, this type of bond is known as a polar bond. This will have a very distinct set of properties than if A has the same electronegativity as B. And I should just quickly. So if A has the same electronegativity as B, the covalent bond is nonpolar. There is no difference in charge between them. They would be held, even though A is bigger, the electron will be held and shared equally. There will be no partial negative or partial positive charge across that covalent bond. And so you have, of course, again, distinct set of properties. So the nature of how those atoms are held together leads to very unique and complex sets of properties. Not all covalent bonds are the same. And this is important when we consider that not only are particles held together differently, but how they come together is different too. Knowing these mechanisms helps us to make predictions about these new macroparticles and their effects on the new system. So what does that mean? So these covalent bonds, these different types of covalent bonds can happen differently. Hydrolysis, recall, is how water breaks covalent bonds. So when water breaks a covalent bond, you're going to, of course, have a different set of molecules than if you go through an oxidation process, where an atom will lose electrons to form an ionic or covalent bond. Of course, you can have reduction reactions where an atom will gain. Now remember, reduction does not mean that the charge will be going. Normally kids think, oxidation, you lose electrons, you must become more negative. No. When you lose electrons, a particle will have more positive than negative charge. Okay? If it has more positive than negative charge, it will have very different consequences. Now, if it's reduced, the particle will have more electrons than positives, which will make it more negative. Okay, so these types of reactions will affect the outcome and their properties. If you've got a system with more positive charge or more negative charge, you're going to have a very different set of chemical and physical properties. And nature has taken advantage of that. Sugar, held together by covalent bonds, is oxidized through glycolysis, through the citric acid cycle. Ultimately, those electrons are shuttled through the electron transfer chain, and oxygen's reduced. Okay, remember, oxidation and reduction are coupled together. You can't have something oxidized if something else is not reduced. So this oxygen provides the driving force. For ultimately the phosphorylation of ADP to ATP. So oxidation of sugar enables phosphorylation. Remember, phosphorylation is the covalent transfer of a phosphate ion to ADP to make ATP. Wow, and then now you have the energy needed to do cellular work. If we look at the particulate nature of this, we can recognize that there are essentially molecular collisions taking place, certain patterns of molecular collisions. These patterns of molecular collisions is what we mean by a process. And this process is driven by the reduction of oxygen. Ultimately, though, these molecular collisions involve enzymes. These enzymes are controlled by genes. And these genes are added upon and manipulated and changed through natural selection. And so we can immediately begin to recognize that genes control enzymes, which can control the process of how energy is distributed in a cell. This is making it necessary for life. Okay. And so a simple physical gene, remember, genes are particles of DNA. Physical nature of genes lies at the bottom of life. It's the molecular biology of life. Combinations of these different outcomes leads to the cellular basis of life. Ultimately, organ systems, individual traits of an individual, traits of a population, traits of a community, of an ecosystem, and of a biosphere. So if you really think about this, changes in the genes, which can be, of course, shuffled around in meiosis, can affect you. If we have a tremendous amount of variation in a system, if there's a tremendous amount of change in a system, you're going to affect the selection of genes. You're going to affect the ecosystem, and that can affect you, how you live in your environment. And so, in summary, I'm going to pop this up one at a time to not overwhelm you. The particulate nature of matter is a model that helps us to predict natural phenomena. When you close your eyes, you can imagine molecules as simple particles, billiard balls coming together. We observe nature at the macroscopic level, though. We see us. We see the effects of those particles. But we can infer them by measuring things, by looking at solution color, by measuring temperature, mass. And we can visualize changes in these solutions. Specific manipulations, changes in how we combine things, allows us to understand the subsequent changes in that macroscopic system. This can help us to confirm our particulate nature of matter. That's what you do in the lab. We're going to do that when we change when we change the composition of our enzyme reactions. We change the color. Biology is hierarchical, built upon the structure, processes, and functions of each level of that hierarchy. There's molecular structure, cellular structure, physical structure of my arm. And it affects changes in those structures and processes affects each level of the hierarchy. The foundation of biology requires molecular collisions, or atoms and molecules that can be modeled as particles. When I close my eyes, I see billiard balls colliding into new macroparticles with a new, unique set of properties. Therefore, changes in the particles and how they combine have the capacity to set off a change in the system, which can ultimately affect each set of emergent properties. and ultimately you. In connection to our current unit, meiosis mixes and matches genes, affecting the particulate nature of the new organisms, which can alter the dynamics of the system. And so, if we go back to our picture, we can now imagine these individuals as combinations of different particles of genes, interacting with different aspects of the particles in their environment. These combined to give the diversity of life. But what we need to do now is study more how those changes can take place. We know meiosis can affect it, but are there other ways?